Welcome back to Viral News, where we keep you up to date with all the latest trends and information related to infectious diseases and epidemiology.
Today's viral topic is the transmission of human immunodeficiency virus, or HIV.
As always, let's first check out our viral vocab. For today's viral vocab, we'll want to know about viral load, which is the amount of HIV genetic material, or RNA, in the blood. And in these two terms, seropositive and seronegative, which are referred to the state of either having the detectable antibodies for the virus as seropositive or not having them as seronegative. The process of going from seronegative or becoming infected with HIV and going to seropositive is known as seroconversion.
Thanks, Mason. Now we're going to turn it over to our colleague Jesse, who's with Dr. Quinn in Uganda.
Thanks, Noah. My name is Jesse Infectious, and I'm here in Uganda with Dr. Quinn, the first author of today's study. Dr. Quinn, what were your objectives and motivations coming into this study?
Yeah, Jesse, I've always been fascinated by HIV-1, the most common form of the disease. And as a scientist hoping to reduce its rate of transmission, my team and I sought to investigate the relationship between viral load and the rate of transmission of HIV-1 in this study.
Great. Now that we know why you're investigating the influence of viral load on HIV transmission, tell me how you went about this study.
Sure. My team and I worked with over 400 families here in Uganda who were discordant couples for HIV-1 infection, which essentially means that one partner was seropositive and one partner was seronegative. It's important to mention that all couples were provided detailed instruction about HIV-1 infection prevention, including condom use. Then, throughout an extended period of time afterwards, we collected blood samples from individuals in each consenting couple to track HIV-1 transmission between couples and also quantify HIV-1 load in each individual.
That made sense. After data collection, what did you do with the data and what did you conclude?
Great questions. We analyzed and organized our data to see if there was a significant relationship between HIV-1 RNA copies per milliliter of blood, or viral load, and transmission rates. We found that at a lower viral load, there is a lower transmission rate, and at a higher viral load, there's a higher transmission rate. This positive relationship tells us that a partner who is seropositive is more likely to transmit the disease to their partner if they have a higher viral load. We also saw no consistent differences between male to female transmission and female to male transmission.
Thanks, Dr. Quinn. Back to you, Noah.
That was interesting.
Agreed. Given the results obtained, it seems to me we're in need of some interventional techniques which reduce HIV viral 1 load to reduce its rate of transmission.
Yeah, this is Viral News. I'll see you next time.
So based off of this video, does anybody have any questions?
Yes. Were there any ethical considerations about this study since it took place in a developing country and their treatment was limited?
Actually, yes. Dr. Angel raised some ethical concerns. Researchers left it up to the HIV partners whether or not to inform their partner that they were positive. Additionally, several hundred people with HIV were observed but not treated. Angel argues that the people of Uganda were treated differently than would have been in a developed country. She believes ethical standards and the welfare of subjects should not be influenced by the political and economic conditions of the region. That means the HIV-negative partner should still be informed regardless. Dr. Angel says this ethical dilemma would only be more of a reason to question whether the research should have been done in the first place. Angel expresses that research conducted in one location should benefit the subjects in that location, but these findings don't benefit the test population. Any other questions?
Yes.
Did the scientists who did the study have anything to say in their defense?
The researchers noted at this time, HIV study participants could not have been easily treated with antiretroviral drugs. The Ugandan government and researchers did not have the clinical capacity to manage treatment as it could cause side effects that the hospitals could not handle. Also, the Ugandan policy was that it was the right of the patient to decide who to inform about their results due to discrimination that could put them at risk, so the scientists couldn't legally inform the test subjects. The researchers note that they did provide HIV testing and counseling, encouraged positive subjects to share with their partners, and provided free health education. They also provided some treatment for other STDs identified. Does anybody know what the impact of this study was at the time and how things have changed since?
Immediate implications from this study were that it raised a lot of alarms about ethics and researching people in developing areas, especially regarding access to medicine. The Declaration of Helsinki, which was a set of ethical considerations when experimenting with humans, was amended 6 months later because of this study and others like it. And then since this study was conducted in 2000, HIV drugs have made a lot of progress. There were drugs at the time that used reverse transcriptase inhibitors, which prevented the copies of HIV RNA into DNA, but they had a lot of side effects. In developing countries with limited health resources, it would have been very difficult to treat these symptoms, and now access to antiretroviral drugs to treat HIV infection have increased by millions of users in developing areas and is less expensive.
These drugs have also been engineered to have fewer side effects, so developing countries can handle them. Also, treatments are more effective because of the antiretroviral therapy, which uses a 3-drug combination to reduce the viral load. The 3-drug types of reverse transcriptase inhibitors, entry inhibitors that block HIV from entering cells, and protease inhibitors that can prevent copies of proteins from being produced can all work together. Overall, now if this experiment was conducted in a developing country, it would be more of an ethical imperative to treat the subjects as there are more resources available.